Well, thank you, everyone, and welcome to Barnett's webinar on RECIST, a disease response assessment tool developed for clinical trials of solid tumor oncology agents. Thank you for joining us today. So let me start with a little bit about myself. My name is Vanessa LaRoche, and I'm a research professional with about 20 plus years of experience in clinical trials. I've served in many roles over the past few years. I've been a research coordinator. I was a monitor for quite some time. I was a program and operations manager at a very large academic medical center on the East Coast. I served on the IRB as a compliance officer for their oncology board. And for the past, say, 5 or 6 years, I've been working in the quality assurance world as a quality manager. I'm currently at a mid-sized pharma company on the East Coast. So at the end of today's session, you should be able to apply the knowledge learned to these tasks. You should be able to discuss the history of the early response criteria and RECIST. You should be able to differentiate between the two RECIST versions, 1.0 and 1.1. You should be able to apply your knowledge gained to calculate tumor burden and determine disease response using RECIST. You should be able to review common trends with tumor data to help develop protocol and case report forms for oncology trials. And then finally, you should be able to review the variations of RECIST and the other response criteria and have a little bit of knowledge on those different variations so that you can determine if any of those other modifications would be beneficial for your studies. And then during this presentation, we'll also complete a very brief interactive exercise just to use the information that we will be covering today. Again, to maximize the value of today's learning, feel free to interrupt me at any time if you have any questions or comments. And again, you can use this either by raising your hand or taking yourself off mute. And there will be opportunities during this presentation where you'll be asked some knowledge check questions, and we can also take questions at that time as well. So let's get started with a discussion on some of the basic concepts behind evaluating treatment outcomes and the history of RECIST itself. As a side note, throughout this presentation, I may use certain terminology interchangeably, such as guidelines versus criteria, cancer trial versus oncology trial or study, treatment outcome versus treatment response, tumor burden versus tumor load, or I might also say follow-up evaluation or assessment versus follow-up visit. So those terms are all interchangeable. If there's any confusion, please again feel free to stop me at any point. So the purpose of an oncology trial is to determine whether a cancer treatment is effective. And assessing changes in tumor burden over the course of a study is one way to determine the effectiveness of that treatment. Tumor burden, or you might also hear tumor load, it refers to the number of cancer cells, the size of a tumor, or the amount of cancer in a patient's body. Tumor shrinkage and time to disease progression, they're often common endpoints used in many oncology trials, but these endpoints are only useful if you're able to systematically measure and compare treatment outcomes. So comparing treatment outcomes, it not only allows for prospective endpoint evaluation in clinical trials, but it also serves as a guide for assessing effectiveness of those cancer treatments. So initial attempts to standardize tumor response assessments, they began in the 1940s, and one of the first standardized response criteria was developed by Sidney Farber at Boston's Children's Hospital, and his team developed a criteria to assess progression in pediatric patients receiving treatment for acute lymphoid leukemia. And in 1956, the team published a formalized set of outcome criteria for hematological malignancies, and that formed the basis of the criteria that's still used today in this therapeutic area. The following decades, other cooperative groups began to define what data was necessary to report in a clinical trial and what constitutes response to a cancer agent. In the 1960s, C. Gordon Zubrod and members of the newly formed Eastern Cooperative Oncology Group, or ECOG, they developed a model for multi-center clinical trials. And then they also argued for the development of criteria for including and excluding subjects, assigning treatment, and measuring response. As a side note, Dr. Zubrod and ECOG were also responsible for developing the ECOG performance status, or you might hear ECOG scale or ECOG score. And if you have any experience in oncology, you will be familiar with that acronym. And we will touch a little bit on ECOG later in the presentation as well. So the first international standardized criteria that was published by the World Health Organization, that happened in 1979. And the WHO criteria, it summarized the minimum data required to allow for comparison across studies. And this criteria defined exactly what constitutes a response, and they came up with 4 classifications: complete response, partial response, stable disease, and progressive disease. So that WHO criteria became widely used, but it also brought to light some deficiencies with the criteria. Some problems included the variability in the way different groups interpreted the criteria, the minimum lesion size numbers recorded, and the definition of progressive disease. Additionally, this criteria, it didn't take into account new imaging technologies, and there were discrepancies identified during independent reviews of reported responses. Investigators and sponsors made modifications to the WHO criteria to address these deficiencies, which in turn resulted in greater variations in how responses were calculated. So it kind of defeated the purpose of having a standardized criteria. As a result of these deficiencies in the WHO criteria, several clinical research organizations, they took it upon themselves to update the criteria, and they published a new standard in 2000, and that's what we now know as RECIST, and that's RECIST version 1.0. But what exactly is RECIST? So RECIST is an acronym. It stands for Response Evaluation Criteria in Solid Tumors. It's a standard designed to provide a clear set of criteria to evaluate progression, stabilization, or the responsiveness of tumors. In other words, RECIST is a standard way to measure how well a cancer patient responds to treatment with a new cancer drug. It's based on whether the tumors shrink, stay the same size, or get bigger. Both the WHO and RECIST criteria, they share the same principles, but RECIST is a simplification of the WHO standard, and it specifically requires that certain lesions be identified as a target lesion so that you're able to track tumor burden over time. RECIST was jointly created by the European Organization for Research and Treatment of Cancer, the National Cancer Institute in the US, and the National Cancer Institute of Canada Clinical Trials Group. So these groups established a RECIST working group to review the existing criteria and to also recommend changes and to come up with RECIST version 1.0. The working group has actually released 2 versions of the criteria over the past few decades. RECIST 1.0, which was published in 2000, and then RECIST version 1.1 was published in 2009. So it's important to note that RECIST version 1 was not retired after version 1.1 was released. So both versions of the criteria are currently being used across the industry. RECIST version 1.0 was first published in the Journal of National Cancer Institute in 2000. And again, it sets forth the rules to define when cancer patients respond, progress, or remain the same over the course of a treatment. Version 1.0 was originally intended for the assessment of solid tumor response in phase 2 clinical trials, but since its publication, the criteria has been adopted across all phases of clinical trials. But with the widespread use of this criteria, again, a number of problems have been identified with version 1.0, very similar to the deficiencies identified in the World Health Organization criteria that came before it. So some of the problems identified with version 1.0, they raise questions such as, could fewer than 10 lesions be assessed without affecting overall response? How should RECIST be applied in randomized phase 3 trials where progression and not response is the primary endpoint? How should newer imaging technologies such as FDG PET and MRI be used? And then finally, how should the assessment of lymph nodes be handled? So again, the RECIST working group convened to address these questions, and they implemented a former overhaul of version 1.0, and they released version 1.1 in the European Journal of Cancer in 2009. And this revised criteria incorporated some changes to simplify, optimize, and standardize the assessment of tumor burden in clinical trials. So we've provided both of these articles in the course material for you. And I do highly recommend that you read both articles so you can get a better understanding of how RECIST works. It's not too bad of a read for the non-technicians, but it's definitely important information that you should have and be aware of.